if you learned about the different secretory glands, exocrine secretory glands, right here. And what else do we need to say? Okay, so. Okay, here, in the duodenum we can try. I can try to go around and find the Meissner plexus in the submucosa, because the actually it's probably easiest to find here, because what does it do? It innervates the glands. So the submucosal plexus is probably much more developed in the duodenum than anywhere else. So let me just go around and try to find one. But I'm gonna go to the 20x objective. If I'm lucky, then yes, there is one. Okay, the Meissner plexus. Here we go. This is one of the ganglion that is part of the Meissner's plexus. Okay, right here, and blood vessels and everything. So there is another one. So. Okay, so this is the Meissner plexus in the submucosa. And again, the reason I tried to find it here, because it should be bigger here, since there are lots of submucosal glands. And now I'm looking for the Auerbach plexus again, so this is the inner circular, the outer longitudinal layer of the muscularis externa, and here we go.
It's hard to see.
There are the ganglia in between. There is one here and the part of the other one there. Okay? So that's the Auerbach plexus. And maybe just blood cells. That's one here. Yeah. And as you know, nerves and ganglia and blood vessels run together. So there is the ganglion and the nerves, and then the little artery, and then the vein. All right. What's the next one? All right, so we're gonna go and try to find Paneth cells in this one, and I move back with the power to the lower power and try to find a nicer region. Here, this is not very pretty. Okay, but don't worry, we will find what we want. All right, here, and we're gonna look for Paneth cells. You see the villi, and that's the same in the duodenum. And here are the crypts, and I'm gonna go to higher power, and they say that there are Paneth cells here, so that there should be Paneth cells. But this is a PAS staining. We will not see the Paneth cells with PAS. Okay. So there are Paneth cells, but this is a PAS staining, so they don't stain. So let's go to the next one. There's
how many more?
We hope that in between the dried mounting material.
This is a lot. And the video is not that great.
Tissue that is actually viewable. Oh, there. This is very bad. The villi are very pretty. This must be the. But the Paneth cells are there. Yeah. Let me just see one clear area. I try to. Oh man.
I'm hungry.
Let's borrow one from the students, because this is just. Can we borrow? This is slide 88. One of you, because this one is just really, really bad. It is so dried out that we cannot see much on it. And there are a number of Paneth cells. All those very dark red granulated cells are going to be Paneth cells. But the mounting medium is so dried out that I just cannot really get in good focus here.
Do you have an 88? Oh, it's crap.
And all these are the large villi in cross section. Nobody has a good 88. We have one good 88. Okay, then let's share it with the rest of the class. Oh, beautiful.
Beautiful.
Like $2?
All right. So if you look at this,
That's like leopard print. I like that.
this is the plica, right? This big, the plica, and then there are the villi, and here you see the villus actually from the bottom to the tip. This is the lumen with some debris in it. Okay, so over here, those are all the villi, and then we will go down here, and in the crypt we're gonna see the Paneth cells, and you cannot miss the Paneth cells. I promise you're gonna get one for the exam.
Good to know. The
All
Paneth
these
cells.
dark red stained cells down here in the crypt, okay? These are the Paneth cells, and down here also. And I even go to one higher, at least the 40x, to see it. If you go to higher magnification and you don't have enough light, just
They look like little flowers.
lift up the condenser, and then you're gonna concentrate more light to a small area, and here you see the Paneth cells in the crypt, so those large granulated cells. What else do we see? We can also see lots of plasma cells and eosinophil cells in the infiltrating eosinophil cells, that eosinophil granules here that we used to show, and the number of plasma cells in this area. So, what? Is there a question? And on the surface, although this is not a pastin because the goblet cells are white, right? We still see, kind of imagine the brush border because the cell surface membrane is so concentrated in this area.